I think the main way to determine what experiments you need to do is to write down what questions you're trying to answer. So once you write down whatever type of question you're trying to answer, if you look in the literature and see, how would I answer this question? What experiments kind of mimic what I want to do, what I'm trying to get at? Once I do that, then I'm like, oh, this is definitely the next step. So when I'm looking at mice, how do I design a orthotopic xenograft model? How do I look at breast cancer development in mice? How do I look at human breast cancer cells being colonized in the lung? How exactly do I look at that experiment? I think the main thing is reading the literature, trying to figure out what experiment I'm trying to do from the literature and seeing how does that fit into the context of what I'm studying.
When you're trying to figure out an experimental strategy, it's worthwhile to remember that even though we think that there are a lot of different kinds of experiments, they actually fall into some pretty straightforward categories. In general, if you can think about like, what are my perturbation tools? What are my measurements that I can make? What's the time scale at which I can make those measurements? And in how many individuals can I make those measurements? And I think it can be really useful to break things down in that way, right? Like, what are my perturbation tools? For instance, you can break the components and measure what happens, overexpress or augment the activity of the components and see what happens, look at natural populations and how they are different from one another in whatever feature you want to look at. And those things map on different systems in different ways. And then ask yourself, what are you going to measure? Are you going to measure a molecular phenotype? Or are you going to measure an organismal phenotype, like a behavior or a developmental character or something like that? And then once you think about that, what's the time scale at which I can make those measurements? Are you going to measure it continuously over time in a single case? Or are you going to measure it statistically at one time over lots of individuals? And like, what are the caveats of doing both of those things in those two ways, right? Like, there's usually a trade-off somewhere between the throughput with which you can do something and the resolution with which you can do it. And then ask, in how many individuals can I make those measurements? Be they individual cells or individual organisms, or that's sort of like, how is the material process, such as, am I looking at single events or am I looking at average cohort either over space and time? That can help you whittle down what techniques are available to you in each one of those cases.
So we have a classification system in my laboratory for 3 kinds of experiments and results. Class 1 experiments are those where either answer is interesting. And they're actually relatively rare to find class 1. Most of the experiments that we do are what we call class 2, where usually only the positive result is meaningful and the negative result doesn't actually move the ball. And it's really important to know the distinction between class 1 and class 2, because if you get it wrong, you start chasing negative results that you think are meaningful that are not, you can really go off in the wrong direction. And class 3 experiments, of course, are those where neither answer is interesting and informative, and they're to be avoided at all costs. What would a class 1 experiment look like? One kind of experiment is to be descriptive. And so that challenge of then deciding which characteristics will we look at, how broadly will we catalog them, will it just be in one cell type and so forth, right? Are all decisions that need to be made, but you can see that the experiments are class 1. It doesn't matter if there's a methylation on this histone at that position. You just want to know if it's there or not, and under what conditions is it there versus not there. So it's all informative, it's all class 1. So it's really important in the planning of every experiment to ask, is this class 1, 2, or 3?
This is going to sound very self-critical. I wish I would have known the extent of my own laziness in terms of doing science, because basically the way I approached grad school was using the tools that have already been developed in the lab and applying them to specific projects. I wish I had been more open to considering new types of measurements and techniques earlier, because the window of opportunity for incorporating new skills into your research repertoire shrinks over time as you try and zero in on a project and get it done in order to graduate. In not being adventurous, I missed out on the opportunity to do the right experiments at the right time. Looking at a given project, like, you can attack it from many different angles all the time. And you basically choose the angles that you think are best scientifically and experiments that you think you can perform. And those are the two variables that you kind of optimize. But it's the second one that's the key, the ones that you think you can perform. If you are exposed to lots of different techniques early on, then you're no longer limited by that and can perform any experiment, especially the one that you absolutely need to further the project at a given time. I think that's really important to keep in mind. So as you broaden yourself out early on, it will help you attack the right experimental questions as you continue in your project.
I think the most important part of picking and choosing from existing techniques or developing new ones is to understand what your investment in doing something new is going to get you, right? And in this sense, what you really want to do is kind of black box the technique, right? You want to think, if I could do this thing, right? And I'm going to black box it. I don't know how you do it at all. But if I could feed this thing into it and get this thing out of it, then I would be able to learn what, right? Like, what would be the great value here? But I'm a big believer in the fact that as scientists, we should communicate openly with one another and share tools and resources, because that maximizes our investment in science as a whole. And there's no reason for us to duplicate efforts if we don't have to. And so in that sense, I'm very reluctant to invest technically in methods that I think would be appropriate for us to use if somebody else is already doing them.
In making progress on a research question, after you're convinced that the system is working and you can make some good measurements, I think it's important to start writing papers in your head, or at least thinking about what set of possible figures would constitute a paper that people would be excited to read. In doing that, it points you to experiments and also holes in existing experiments that you can follow up on and fill over the course of your graduate career. And so I recommend it as a tool in terms of conceptualizing your project and its direction.